Hi everyone, in this lecture we will be continuing with our studies and preparations for the British Biology Olympiad, specifically looking at biological molecules and carbohydrates as the main focus unit under that large topic. In this video we're going to look at different types of monosaccharides, proceed into looking at disaccharides and how disaccharides are formed by glycosidic bonds. I do have two other videos, part 1 and part 2, before this video, and I think it's very important for you to visit those videos to gather prerequisite knowledge to understand and process the content of this video. So if you haven't watched that already, please go do so. And so in this video we're first going to focus on the other types of monosaccharides. So far the focus has been on hexose sugars because we've been looking so much at glucose, but monosaccharides can be more broadly classified by the number of carbon atoms that they contain in their rings. Triose, for example, they contain 3 carbon atoms, and an example of this would be glyceraldehyde, which plays an important role as a sort of an intermediate molecule in the metabolic pathway such as glycolysis. We have tetrose, which also contains 4 carbon atoms, an example being erythrose, which is involved in carbohydrate metabolism and biosynthesis pathways. We also have pentose, and this is something that we should have looked at earlier on, and this contains 5 carbon atoms, and it's very important biologically in that there are mainly 2 pentose sugars that are especially significant, and this is the ribose sugar, and this has the chemical formula of C5H10NO5 and it's a component of RNA and ATP. We also have deoxyribose, which pretty much has a very similar looking formula to ribose, except for the fact that it has 1 less oxygen, and it is used for DNA. Just as a recap, the difference between these two is that ribose does have an OH hydroxyl group in the place where deoxyribose has a single hydrogen group. And the presence of this oxygen, or the absence of it, can definitely contribute to the overall polymer stability. And the RNA world hypothesis, which hypothesized that ribose can also form enzymes such as RNA can also form enzymes such as ribozymes and thus can self-replicate. Right. Next, we're going to look at different types of disaccharides. Disaccharides are basically carbohydrates that are formed when 2 monosaccharides can be joined together by a bond. And common biological disaccharides include sucrose, maltose, and lactose. And as you can see, we have different monomers or different monosaccharides that make up this disaccharide. For sucrose, we have glucose and fructose. Lactose, we have galactose and glucose. Maltose, we have glucose and glucose. And although the disaccharides do consist of 2 monosaccharide units, they differ in the types of monosaccharides that are involved and the type and the position of the glycosidic bond that is linking them. You can kind of see that visually. We'll be talking about this later on, but there is a difference in the glycosidic bond that is linking the monosaccharides. Right, okay, so more about the glycosidic bond. Glycosidic bond is basically the covalent bond that joins together these monosaccharides in order to form polysaccharides. And a glycosidic bond forms when the hydroxyl group of one monosaccharide and the hydrogen group of another monosaccharide are removed altogether in order to form a molecule water. You've probably heard of this reaction. This is the condensation reaction, and it's also known as an anabolic reaction. It's a type of anabolic reaction wherein we have a lot of small molecules gathering up to form a larger sort of molecule by the removal of water. And it's also the opposite of a hydrolysis reaction. And the remaining oxygen atom that remains in this area right here, it's going to act as a sort of bridge between the 2 monosaccharides, linking them together through their carbon atoms. Now, this is very important because it forms the basis and a strong sort of stabilization bond that forms disaccharides and polysaccharides from monosaccharides. Right, so this is pretty much a diagram that sums up the formation of maltose. We have 2 monosaccharides. We have 2 d-alpha glucoside molecules, and as you can see, we are removing water by the condensation reaction to form glycosidic bonds. The OH group, the hydroxyl group of 1 monosaccharide, removed with another hydrogen from another monosaccharide, removed all together to form water, and this in turn forms a glycosidic bond linked by a single oxygen atom to form a disaccharide, which in this case is maltose. I think it's very important for me to kind of clarify one thing. I think it might be confusing for you guys, when you're seeing this diagram, because you could have like presumed that maltose is made of 2 alpha glucoses, and this molecule doesn't necessarily look like an alpha glucose, or rather looks like a beta glucose because of the orientation of the hydroxyl group. But I think it's very important for us to clarify that when we kind of say that maltose is made of 2 alpha glucoses, and that this bond is a 1,4 alpha glycosidic bond, we typically refer specifically to the sections that are the carbon atoms that are involved in the bonding. We need to know that this atom or carbon atom right here, carbon 1, and the respective hydrogen and the OH groups that are attached to it, this is kind of free moving, so it can mutarotate, and so it doesn't necessarily have to be in the formation of an alpha glucose at all times or a beta glucose at all times. And again, that is, I think, something very important that we have to note when we're seeing diagrams like this, where it can be confusing whether this is an alpha glucose or a beta glucose, just for you to note. But again, maltose is made from 2 alpha glucoses linking together via a glycosidic bond. That's 1,4 glycosidic bond. Right, okay, glycosidic bonds. I kept on mentioning this. Glycosidic bonds are named according to the carbon atoms that are involved in linkage. And for example, if we were to look at a 1,4 glycosidic bond, this would mean that the bond is forming between carbon 1 of the first monosaccharide and carbon 4 of the second monosaccharide. We read from like this clockwise direction. This is carbon 1 of the first monosaccharide, and this is carbon 4. We had previously an OH group right here, and we had another OH group right here, and we can see that here the H has been removed and OH has been removed entirely, so now this single oxygen atom is really facilitating that covalent bond right there. So the numbering system is very important, because in our next video we'll be also looking at different types of glycosidic bonds, specifically the 1,6 glycosidic bond, and that's going to be a very different one where we have bonds forming with 1 and 6. So again, that's a lesson for another day, but do remember the naming rules specifically in order to understand all types of bonds.